it, and then it comes down into your throat, and you might just simply swallow it. You're breathing in. Okay, so if we look at the back of the nasal cavity, we have opening into a structure which is referred to as the pharynx. So the pharynx is the next structure. And when we talk about the pharynx, this is the common opening to the respiratory and digestive systems. So there's different parts of the pharynx. So starting up at the nasal region, this is what is referred to as the nasopharynx. All right, so this is the back of the nasal cavity. Something really interesting is found at the nasopharynx, and that is what is called the eustachian tube or the pharyngotympanic tube. So if you look at this word, that's the pharynx. And what do you think the tympanic refers to? Your eardrum, right? So what is this commonly called? What do you commonly hear it being called? You say in babies they have clogged tubes, but specifically these are also called eustachian tubes. Have you guys heard that word before? This is what I learned. So eustachian tubes are the same thing as those pharyngotympanic tubes. So if we look at this diagram right here, you'll notice that there is an opening. Here they call it the auditory tube, so we'll also call it auditory tube. Okay. So this connects the back of the throat with your middle ear. So your middle ear contains those three bones, the stapes, the incus, and the malleus, and it is generally filled with air. But sometimes you can get bacteria that come in through your nasal cavity up into the eustachian tubes, and you can actually get a middle ear infection. So sometimes when you're sick, you'll get that, and then your ears will feel full, and then it will be much more difficult to hear because if they're filled with fluid, then the bones do not work as well at transmitting the sound waves from or into the inner ear. So this sometimes gets blocked, right? Specifically in babies, it seems that they get a lot of middle ear infections. And interestingly, there has been a push to not give antibiotics to babies and young children so frequently. And so sometimes it's really hard to get antibiotics for that middle ear infection. One thing that they have discovered is that simply the act of breastfeeding, so suckling, the movement of the tongue and the mouth when a baby suckles, when a baby nurses, actually appears to help keep that eustachian tube open. And so it is less likely to get they think that babies are less likely to get middle ear infections if they nurse, rather than if they simply take their nutrients via a bottle. The other thing that you'll notice that is up in the nasopharynx is that pharyngeal tonsil. And I mentioned it previously when we were talking about the lymphatic system, because this is part of the lymphatic system. And do you remember what the other term for that pharyngeal tonsil is? Adenoids, right? So your adenoids would be located right up here. And remember that the function of the tonsil is actually to capture microbes and present the microbes to your leukocytes so that they can learn to identify them, so that if they do become a problem, that you can learn to fend those off more quickly, okay? Okay, so this right up here, this is the nasopharynx. When we move back down here, this region right here is the back of the throat. And so that is called the oropharynx. Oops, I spelled that wrong. Okay, so this is the back of the throat. And your big tonsils, the palatine tonsils, are what sit there. Okay. The back of the throat, the oropharynx actually changes from being pseudostratified to stratified squamous. So now we have stratified squamous tissue. So you can think about anywhere where we might have lots of friction, we're going to have stratified squamous. So in the back of the throat, when we're swallowing food, sometimes it scratches the back of the throat. So it'd be really bad to have a simple layer of epithelial tissue. So we need to have stratified so that it doesn't bleed, right? It protects against friction. Then moving down, we have the laryngopharynx. The laryngopharynx. So this refers to where it is going to switch from being the pharynx into going into the esophagus and then into the trachea. So this is a switching point for air and food. So you want air to go into the respiratory tract, you want your food to go into your digestive tract. And so this is where that occurs. And so at the laryngopharynx, we have an opening called the glottis. So this is the opening into the respiratory system. So a glottis is actually the space that the air enters into the respiratory system. So if we look at our diagram, this would be my laryngopharynx right here, right? This space is what is referred to as the glottis. And you'll notice that there is a structure that can sit upon the glottis, which is referred to as the epiglottis. So the epiglottis just means upon, epi means upon the glottis. And so the epiglottis is what functions to close. So we can say the epiglottis. This is what closes off the glottis. And it is a mechanical valve. So what that means is that something has to push against it in order for it to close. So it tends to push against it. Okay. So when we swallow food, we'll talk about this, but a food ball or bolus can actually push against that epiglottis and cause it to fold over. And the epiglottis is very flexible because it is composed of hyaline cartilage. So it is kind of like the same cartilage as your outer nose. It's your hyaline cartilage. The most common cartilage that you have in your body. It's always drawn in as blue, cartilage is. So you can see that there's other pieces of cartilage, which we'll talk about in a minute. Okay, now there is one other mechanism by which we can close off that glottis, and that is by increasing abdominal pressure. So there is a pressure difference when we do what is called the Valsalva maneuver. And this is a maneuver. This is where you have abdominal straining. So this tends to close off the respiratory system. So like if you're going to pick up something, right, really heavy, you press, your abdomen contracts, right? Hopefully you're using your core. So your abdomen contracts and you pick it up and you feel like your respiratory system gets closed off, right? Obviously, this could also be when we go to the bathroom. Sometimes we have abdominal straining, and that can also close off the glottis and prevent stuff from getting into your respiratory system. Okay. This is the diagram I was looking for. So this here you can see the nasal concha and how they greatly increase the surface area. This is all lined with that mucosa respiratory membrane. And then you can see the nasopharynx and the eustachian tube. So this is the opening to the eustachian tube. And then this is the pseudostratified epithelial tissue. So the location of the nuclei make it look like that it is more than one layer of cells. And then if you look really carefully, you can see that on the surface of the cells are the cilia. And so these are just cytoplasmic extensions that are covered in mucus, and they can actually move and force the mucus down or away from your nose and into your throat where you can swallow it. Goblet cells are specifically the type of cell that produces the mucus when we look at this tissue. Okay, so we already talked about the pharynx. So this is a diagram from the new textbook that shows the different regions, right? The nasopharynx, oropharynx, and the laryngopharynx. So the next structure we're going to talk about is the larynx. And this is sometimes referred to as the voice box. So air actually has to travel through this to get into the trachea. So it's a passage for air, passageway. Right? But we can also use it for vocalizations, right? So we can actually create sound waves as air moves out of the lungs. So this is where we have the vocal cords. Sometimes these are also referred to as vocal folds. The true vocal cords are white in color. And you can actually cause the vocal cords to be long by constricting and changing the shape of the larynx. And you can also cause the vocal cords to be shorter, and that is what helps to change the pitch of the sound. So remember, we talked about sound waves when we were talking about hearing in 232. We talk about hearing, and the sound waves have to do with the frequencies and the amplitudes. And so the higher the amplitude, the louder the sound. A high frequency is a higher pitch, and a low frequency is a lower pitch. So what we're going to do is I'm just going to show you a little video that actually looks at the vocal cords and shows when they put a microscope down, and not a microscope, but actually a recorder into the vocal cords, how they can actually see how they change shape and how they produce sound.
I'm going to show you live video of a singer's vocal cords as she sings. You may want to sit down. Hi, I'm Chuck Gilmore with Power to Sing. Many students feel singing is a great mystery. They don't understand how their voice works, why it works the way it does, and what's involved with singing. It all begins with the vocal cords. Specialists look at your vocal cords with a flexible or a rigid scope. The flexible scope is a small cord with a camera at the end of it. It's inserted through the nose and extended down the throat to see the vocal cords. The rigid scope is like a large pen with a camera at the end. It's used looking into the mouth and throat. Both images look like this. This can be confusing because the vocal cords are horizontal in the throat, like this. They are not vertical, like this. When the scope sees the cords and projects it on a monitor, they look like they're vertical or up and down. Vocal cords, sometimes called vocal folds, are located in the mid portion of your neck, about halfway down, between the top and the bottom of your neck. There are two vocal cords, and they're connected in the front of your neck and open and close in the back, towards the back of your neck. They're located inside the voice box, or larynx. The cords sit at the top of your windpipe, or trachea. Men can feel the top of their voice box, often called the Adam's apple. Women have the same thing, but it's smaller and harder to feel with your finger. Your vocal cords are just behind the Adam's apple. Now, women can sometimes feel their Adam's apple by placing their finger here and gently pushing in on their neck. And about the first lump that you feel is the top of the larynx. About halfway down your neck, you feel that small bump. That's your Adam's apple, ladies. And your vocal cords are just behind it. I can feel a small V right in the center of the top of my Adam's apple. That's the part of it. Now, at birth, our vocal cords are about the same length, approximately 2 millimeters. By age 20, women's cords are about 10 millimeters and men's 60 millimeters in length. The change for men is much greater, and this causes a deepening of the voice and can make singing during the vocal change much harder. Vocal cords are tissue and are three-dimensional. That means they have length, width, and depth. Just like other tissue in your body, there are different layers of tissue. Vocal cords have three primary layers: muscle, ligament, and mucosa. It's the slick outer layer of the vocal cord. The vocal cords open when we breathe and come together when we make noise with our voices, and are covered up when we swallow. A type of lid called the epiglottis closes over the vocal cords when we swallow. Sometimes fluid gets through and we cough to clear it out. When air passes through our vocal cords as we speak or sing, they create a series of sound waves, which we can hear. This series of sound waves we call vibration. The vocal cords create the initial vibrations, which is the initial tones we hear that begin the sound we make, speaking and singing. Now watch and listen as these vocal cords create the vibrations that become our voices. Do you know your vocal type? I'm not talking about whether you're soprano, alto, tenor, or bass. Your vocal type helps you understand what your vocal cords tend to do when you sing. For example, if you are a light chest, no chest vocal type, that means your vocal cords are not closing as firmly as needed. With each vocal type, your vocal cords tend to do specific things as you sing. Once you know your vocal type, you can begin eliminating weaknesses in your voice and get the singing voice you want. Visit powertosing.com.
Okay. So the length of the vocal cords can change, right? So the longer the length, the thinner they are, the higher the frequency, and then the thicker and the shorter they are, the lower the frequency. And interestingly, birds are really interesting because they can sing more than one note, right, at a time when they're actually singing. So they actually have what is called a syrinx instead of a larynx. So they actually have two openings, and so that allows them to sing two different notes, and so they can have really complicated sounds. Some people say that they can. Have you ever heard of that? That some people claim they can sing two different notes at the same time. Very strange. Okay, so if we look at the structure of the larynx, remember that it is a structure that is protected by the hyaline cartilage. And so we have the Adam's apple, which is actually that thyroid cartilage. And the reason why it's called thyroid, because your thyroid gland sits right there up on top of it when you dissect the neck region out. And you'll also notice that there's a bone there, and that is called the hyoid bone. And the hyoid bone is a really interesting bone because it kind of just floats in your neck. It connects to the tongue, and it also connects to this cartilage, but it doesn't really connect to any other bone. And so it's it's a kind of a strange bone. It is actually believed to be one of our gill arches. So when we have gills when we first start developing, and fish also have these bony structures that support the gills. And so this is kind of like a remnant of a gill arch. And then the vocal folds or cords sit here. And so when we press air out through them, we can create sound waves. Are there any questions about that? Yeah. Okay. So the next structure we're going to talk about is the trachea. So the trachea, we're just moving down into the respiratory system, right? So this is the next structure after the larynx. And when we look at the trachea, we now move to that pseudostratified. So we're back to the pseudostratified, ciliated. No, spelled right, ciliated epithelial tissue. So generally, we don't want things that are going to scratch coming down into the trachea. So the trachea produces mucus, lots of mucus. But instead of moving the mucus down like we had in the nasal cavity, we move it up to the throat. So the cilia move the mucus against gravity back up to the throat. Now your trachea sits anterior or in front of your esophagus. And so you can actually feel it, right? So you can feel the trachea and you can notice that it has rings in it. And so we'll say that it is supported by rings of hyaline cartilage. Now the trachea is going to branch into what are called bronchi. And these two bronchi go to the right and the left sides of the lung. So right and left lungs, we have two lungs. So the right and the left lung, so it's separated. And then what's going to happen is that these bronchi are going to turn into a respiratory tree. So they're going to branch and branch and branch into smaller and smaller vessels. So they branching continues until you get to what is called a terminal bronchiole. So that means the end of the bronchi, right? So a small bronchi is called a bronchiole. So that's B-R-O-N, bronchiole. These are super small, like they're like 0.5 milliliters or millimeters. So 0.5 millimeters in diameter. So they're super, super tiny, right? And then what's going to happen is at the end of these terminal bronchioles, we're going to have the alveoli. So it would look something like this. We have the bronchi. We branch into smaller and smaller vessels until at the very end, we're going to get to the alveoli. So that is my alveoli. Or actually singular would be alveolus. Put another one on here. So the alveoli is the only place within this whole respiratory system where gas exchange actually takes place with the blood. So remember that most of our respiratory system is just the conducting zone. So if we look at a diagram of this in your book, you can see this is actually a cross-section through the trachea. So you can see the hyaline cartilage and then the layers of connective tissue, and then we see the pseudostratified epithelial tissue with the cilia on it. Okay. So the next thing we're going to talk about is we're going to talk about the characteristics of the respiratory membrane. And so the respiratory membrane would be the alveoli. And so if we look at the characteristics, the respiratory membrane must be moist. So this is actually a constraint because we actually lose a lot of water from our lungs. Like, you know, when you sit in a car on a cold day and you start to fog up the windows, that is all water that's coming out of your lungs, right? So we lose water across the surface. But it must be moist because this allows oxygen and carbon dioxide to diffuse. And we'll talk a little bit more about diffusion in a little bit, but they have to be dissolved in water in order for them to just passively move across this membrane. The membrane also must be thin. So instead of being stratified, it is actually a simple, meaning a single layer, squamous, meaning flat, epithelial tissue. So it looks really like this. The other thing that it has to have is it must have lots of surface area. Because the more surface area, the more oxygen and CO2 can diffuse more quickly, right? If we look at the lungs of like amphibians, like frogs, they have a lot less surface area. There's a lot less branching of the lungs. And then the alveoli have a lot less surface area than, say, do reptiles or mammals. So as we see the evolution of vertebrates and land organisms, they actually get more complicated lungs. So they have a lot more surface area. And this is all has to do with what is referred to as Fick's law of diffusion. So this is actually from physics. You probably might learn about it in chemistry. And this is how we describe how these characteristics affect diffusion. And so we have what is referred to as D, big D, which is my diffusion rate. This is equal to the surface area times, and we have another thing that we haven't talked about here, and this is the concentration of the concentration of gases in solution. So I'm going to put times little d times, actually not little d, we'll just times it, delta P, that's my partial pressure of oxygen, for example, divided by little d. So surface area, that's pretty easy to remember, SA. This little d is distance. So the smaller the distance, the greater the rate. When distance becomes larger, then diffusion is going to decrease. Okay. So if we look at delta P is the difference in the concentrations of gases. Okay. So delta P, sorry, partial pressure. This is called partial pressure. So it could be the partial pressure of carbon dioxide. I put oxygen up here. So if there's a big difference between inside the lungs versus the blood coming to the lungs, then you're going to get diffusion happening more quickly, because remember that diffusion always goes from a higher concentration to a lower concentration. So if that concentration goes to 0, if there's no difference in the air that's coming in versus the blood that's coming in, then diffusion is actually going to drop to 0, right? And so that's an important thing to remember, is that diffusion will drop to 0, right, if there's no difference in the partial pressures. And we're going to come back to this when we start talking about gas exchange in a little bit. Okay, so this is my diagram, my image of the alveoli. And so one of the things that you'll notice is that these are blind-ended sacs, and they're tiny. And so one of the problems is getting the oxygen or the gases out of the lungs, so the carbon dioxide, after gas exchange takes place. And so we have a problem in that there can be a lot of what they call dead air left in these spaces, in these alveoli, that are going to prevent new air from coming in. So we'll talk about ventilation in a minute as well. You'll also notice that the capillaries surround the alveoli, and so these are the capillaries that are in the pulmonary, that the pulmonary vein carrying, or excuse me, pulmonary artery carrying blood in and pulmonary vein carrying blood out. These are the capillaries of those. So these are our pulmonary, the pulmonary vessels. You'll also notice that when you look at the tissue, so this would be like a cross section through it, you can see that there's lots of surface area. So if I take a cross section through one of those alveoli, you can see that the membrane is thin and that there is a large surface area. So there are a type of cells that are important in the alveoli that don't function in actual respiration. And these are what are referred to as the type 2 cells. So the type 2 cells produce a substance called surfactant. And what this surfactant does is it decreases the surface tension of water. So these membranes have to be moist, and sometimes what happens is that the water molecules are so attracted to each other that they create the surface tension that causes the alveoli to collapse. So without the surfactant, the water pressure, the water tension between molecules, the hydrogen bonding, would actually cause the alveoli to collapse. And so the reason why we know this is so important is that in premature babies, these type 2 cells are not producing surfactant because they are so premature that the lungs were not mature. So when we look at what is called the infant respiratory distress syndrome, so this is IRDS, infant respiratory distress syndrome. This is typically caused by no surfactant. So their alveoli are collapsed in on themselves. So does anybody know how they would treat this? How would you maybe think about treating it? Steroids would decrease inflammation, but it wouldn't actually cause the alveoli to expand. How do you get, if somebody's lung is collapsed, how do you get air back into it? You ventilate it, right? So you can do positive pressure ventilation. So positive pressure ventilation just means that you give them air. So this is actually going to increase the airflow into the alveoli to open up those alveoli, and then you give them synthetic surfactant. So like you would put that in the air. And so that's how you would get the synthetic surfactant down into the alveoli. So when premature babies are on ventilators and their lungs aren't quite ready to work, this is probably the reason why. You'll also notice that there are macrophages located in our alveoli. And so anything that escapes being filtered, once it gets into the lungs, it's actually going to start to, those macrophages are going to start to eat anything that gets down in there. So that would include tar from cigarette smoke, for example. It would include like maybe asbestos fibers that get stuck in the lung. Those macrophages are going to destroy them. And then hopefully it would include bacteria. The type 1 cells are simply the cells that make up the alveoli. So the type 1 cells are just the cells that make up that simple squamous epithelial tissue. The type 2 cells have a different structure. They are more cuboidal, they're not thin, right? And they're the yellow ones in here that are producing the surfactant. Okay, so let's talk about the lungs, the anatomy of the lungs. So the lungs sit in the thoracic cavity. They sit on either sides of the heart, right? So they are protected by the rib cage. They sit on top of the diaphragm. So the diaphragm is our respiratory muscle, and it is skeletal muscle, and we'll talk about its function in causing ventilation and allowing for ventilation to occur. So one of the things that's interesting about the lungs is that one lung is actually smaller than the other. So your heart actually sits over to your left side of your thoracic cavity, so that when you put your right hand on it, you don't generally put it like, you're not like putting your hand right over here. So just think your hand is over your heart on the left side. And so notice that this is actually the right side of the diagram, and this is the left side of the diagram. So it's as you're looking at the person, right? So this is the left side as you're looking at a person. It's their left side. So it's always from the perspective of them, right? So the left lung has 2 lobes, and the right lung has 3 lobes. And then this little indentation is referred to as the cardiac notch, right? So the left lung has 2 lobes, right, and the cardiac notch. So it is slightly smaller in its surface area. And then your right lung has 3 lobes. The diaphragm actually kind of sits right here. When it's relaxed, it's kind of a dome shape, so hence kind of why they're shaped this way on the bottom. Now, the lungs are actually encased in a membrane, and we have what is called a serous membrane. And if you learned about the heart, the serous membrane that surrounds the heart is the pericardium. But does anybody know what the serous membrane that surrounds the lungs is referred to as? It's not pericardium. Anybody know? It is called the pleurae. And so we have 2 layers. So the outer layer is called the visceral pleura. And this connects kind of to the ribs and to the outer body cavity, right? So this is the outer one. Think viscera just means, oh, excuse me, that's the inner one. Sorry, so this is the inner one. Sorry, this is the inner membrane. The outer membrane is the parietal. The reason why I remembered that is because visceral means it's on the viscera. So we have the visceral pericardium on the surface of the heart. The parietal pericardium, or the parietal pleura, would be the outer membrane. And then we have what is called the pleural cavity. So that's the space between those two membranes, and this produces fluid. Right, so it's fluid filled. And this decreases friction. So your lungs are able to move, they're able to expand and contract, and there's no friction between them and the other structures. This fluid is actually really important also because the pleural space, the pleural cavity, we'll talk about this, but it has a negative pressure in it. So our pleural cavity actually causes the lungs to kind of sucks the lungs, the negative pressure sucks the lungs, and it keeps them expanded, and it keeps them from recoiling. And so sometimes that pleural cavity can get air in it, and that is a really bad thing, like if you have a chest wound, because if that air gets in there, then it's going to cause the lungs to collapse. And so that needs to stay at a negative pressure. So we'll just put, for right now, this is a negative pressure. Talk about that. Okay, so if we look at a diagram, we can see these, the visceral pleura, the innermost, the parietal pleural, the outermost, and then that space which would be filled with fluid. You can also see the diaphragm sitting in this image. There's also muscles that go between ribs, and so these are called the intercostal and the external, so the internal and the external intercostal muscles. And these allow us to expand our rib cage when we breathe in deeply. So it lifts the rib cage. And then we can also cause our rib cage to depress, for example, when we exhale. So those intercostal muscles also play a role in ventilating the lungs. So when we talk about pressure in the lungs, we can talk about the atmospheric pressure, because this is the same type of pressure that we're talking about. So the atmospheric pressure at sea level is about 760 millimeters of mercury. So this is measured in millimeters of mercury. Now, we can also talk about the pressure inside the lungs. And so we can talk about the interpulmonary pressure. So this is the air pressure inside the lungs. Now, this differs depending upon whether we want to inhale or exhale. So that interpulmonary pressure could go to 759 to 761 millimeters of mercury. Okay, so it can vary. And so what we have, what is referred to as negative pressure breathing. And this is different from other organisms, like even like reptiles. they actually have to force air into their lungs by constricting muscles that push air against pressure and cause the lungs to expand. But we have negative pressure, which means that there is a negative value, and that actually draws air in. So during inhalation, the interpulmonary pressure drops. So this decreases, and this causes air to be drawn automatically into the lungs. So that's what happens. Inspiration is the same thing, to inspire, to bring in air. It's the same thing as the inhalation. You might see them used interchangeably. When you're on a ventilator, this is not how it works. So if I'm on a ventilator, then air is being kind of forced into my lungs and causing my lungs to expand. But the negative pressure, our normal breathing, is done in this fashion. Okay. So this actually has to do with differences in volume and pressure. So as lung volume increases, this actually causes a decrease in the pressure. So if you think about having a balloon, for example, and you blow it up, if your balloon can expand more, then there's actually going to be, and it's not completely full, then there's can actually be a decrease in pressure. And then if you like press on your balloon and try to get all the air into one part of it, then that's going to actually increase your pressure. So as lung volume decreases, then we have an increase in pressure. This actually has to do with the location of and what the diaphragm is actually doing in our body. So when we look at our lungs, so if I drew my lungs right here, the diaphragm sits like this normally, and it's pressing up, and it's actually causing there to be a decrease in volume. So this would be like low volume lungs. So then there's an increase in pressure. So this is what actually happens during exhalation. And this is where the diaphragm relaxes and pushes against the lungs. So that's the diaphragm relaxed. So when the diaphragm contracts, it actually flattens out. So my diaphragm would contract, and then there would be more space for my lungs to sit. And so I would actually get an increase in the volume of the lungs. So I have more space for the lungs to sit, an increase in the volume. So I'll just put that. This is inhalation. The diaphragm contracts and flattens. Okay, so this actually decreases the volume. Oh, sorry, increases the volume and contracts. So this would increase the volume, decrease the pressure. Okay. So normally, sometimes we don't use the diaphragm to breathe, and so we breathe really shallowly. But when we do use the diaphragm to its fullest, and we also use the rib cage, when you inhale, so if you take an inhalation, the diaphragm contracts and the rib cages expand, and so that increases the volume of your lungs. So they're the air pressure, the atmospheric pressure just automatically causes air to come in. And so then when I exhale, my diaphragm would relax up, and I could actually forcefully exhale some more by contracting my intercostal muscles and causing my rib cage to depress. So if I'm going to breathe, you could breathe in, just visualize the diaphragm breathing in, visualize the diaphragm flattening out. And then when you exhale, visualize the diaphragm forming a dome and then contracting your ribs even more to forcefully exhale. So we're going to be doing some of this, measuring what is referred to as your tidal volume, the volume of air that moves out and in and out of the lungs when you normally breathe. And then we're going to be looking at how much extra you can inhale and how much extra you can exhale during breathing. And so that's one of the things we're going to be doing in lab this week. So this is backwards from the way that you might think about it. Because when I think about inhaling, it feels effortless, but that is when the diaphragm is contracting. And then when I think about exhaling, that feels more contracting, but that is when the diaphragm is relaxing. Okay, so you might want to sit down and just think about this a little bit to kind of get it solidified in your mind and breathe and visualize it actually happening. Now, one of the interesting things happens when our diaphragm starts to spasm. And what does a spasming diaphragm produce? Hiccups, right? And so then you know all of a sudden that diaphragm is spasming and it's causing air to move up, and it causes the glottis to kind of snap shut. And so that is what gives you the hiccups. And so spasming of the diaphragm is the basis for that.
So then are all the other tricks and myth, like getting scared and stuff, when really you can just sit there and flex?
No, you know, I'm not sure that, so it's the spasming of the diaphragm is involuntary. So it's not like you could think about it. You might be able to breathe deeply. And I think maybe breathing deeply and holding your breath. So if I'm inhaling and then I hold my breath, that's going to keep my diaphragm relaxed. And so that might stop it from spasming. So the whole inhaling and holding your breath makes sense to me. The way that I get rid of hiccups, though, is kind of weird in that I drink a glass of water upside down. Yeah. Right? So from the back of it. So you go like this, and I don't know, it just works every time for me. Right? So whatever works. Okay, so we will stop there for today. And we'll continue talking about respiration on Wednesday. And then we're actually moving to the digestive system.
Well, I'm going on vacation soon, so it'll be worth it. It'll be worth it. Yeah. Yes. Bye. Are you staying?